Image. The neutral red assay uses a dye that healthy cells will take up, becoming red in color. View more. Credit British American Tobacco. New research reveals that, unlike cigarette smoke, vapor from the tobacco heating product THP-GLOTM has little or no biological effect on cells. THPs, like GLOTM, heat rather than burn tobacco and so have reduced numbers and levels of toxicants and emissions compared to cigarette smoke and therefore have the potential to be much less harmful. Scientists at British American Tobacco conducted a range of tests comparing the impact of cigarette smoke with that of GLOTM vapor on cells in the lab. Cigarette smoke produced a clear response in all the tests used, confirming smoke is toxic and reduces cell viability, causes oxidative stress, gene mutations and the promotion of tumors. In contrast, the vapor produced by GLOTM failed to elicit a response at doses equivalent to or higher than the cigarette smoke, demonstrating it is not toxic and doesn't cause mutations. We have produced a comprehensive suite of scientific tests and data that clearly demonstrates that GLOTM has lower biological activity compared to cigarette smoke, said Dr. James Murphy, head of reduced risk substantiation at British American Tobacco. GLOTM induced either no or marginal responses in all tests conducted, he said. This comprehensive assessment is part of a science-based approach that we have developed to assess the reduced risk potential of THPs and other next-generation products relative to smoking cigarettes. We believe that such an approach is essential to communicating to consumers and regulators that the available information on our products is based on sound, evidence-based science, he says. How the tests were done. Puffing robots were used in the lab to produce smoke and vapor. Cells, including human cells, were then exposed to the smoke and vapor and the impact on the cells compared to the impact of air. Cell viability. How healthy are the cells? The cells tested included human airway cells. These are obtained by taking a sample from a human nose. The cells are placed in a chamber that allows them to be nourished from the underside and exposed to air or vapor or smoke on the other side, much like cells are exposed to aerosols in the lung. The health viability of the lung cells was tested by observing how effectively they can absorb a special red dye. Healthy cells are very good at incorporating the dye into their structures and so appear red. Figure 1. Unhealthy cells lose the ability to take up the dye and so change in color from red to light pink. Figure 2. Cells exposed to smoke got lighter in color as the cells lost the ability to absorb the red dye. In contrast, GLOTM vapor remains red in color, indicating that the vapor is substantially less toxic to cells. When all the cells exposed to smoke had died, those exposed to GLOTM vapor were still 97% viable. Genetic mutations and tumor promotion. In other tests, bacteria were used to determine whether smoke or vapor causes mutations in DNA, the cell's genetic material. Mutations in genetic material are important because they could be involved in the development of cancer. Scientists exposed special strains of bacteria to smoke or vapor and then observed whether there were any changes to the bacteria's DNA. Exposure to smoke caused mutations in a dose-dependent manner. The higher the dose, the higher the mutation rate. Exposure to GLOTM vapor did not cause any mutations, even after hours of continuous exposure, comparable to the results obtained when cells were exposed to air. In other tests, the ability of vapor from GLOTM to cause the development of tumors was compared to that of cigarettes. Results show that even at very low doses, cigarette smoke caused tumor promotion whereas GLOTM vapor did not. Many tests at once. A method called high-content screening was used to test different aspects of the impact of smoke or vapor on human lung cells at the same time. Fluorescent dye molecules are attached to different parts of the cell structure, allowing scientists to observe any changes in the cell structure or function. Cigarette smoke was positive in all tests, but comparable doses of GLOTM vapor showed little or no activity, suggesting it is substantially less toxic to cells. Taken together, the results of all these tests form the basis of a comprehensive dossier of scientific data that lay the groundwork for establishing this product's reduced risk potential compared to cigarettes. The results are published today as part of a special issue of the journal Regulatory Toxicology and Pharmacology. Disclaimer: AAAS and Eureka Alert are not responsible for the accuracy of news releases posted to Eureka Alert by contributing institutions or for the use of any information through the Eureka Alert system.